Welcome back to the conversation. We're talking about research into metals and the immune response with Val Culotta, a professor in the Department of Biochemistry and Molecular Biology. Val, in your lab, you're examining how manganese affects the immune response to pathogens such as infectious yeast and bacteria, including the Lyme disease pathogen. Why manganese and why the worm?
Well, for decades now, we've been studying why cells need manganese, how it protects cells against harmful effects of free radicals, the so-called reactive oxygen species that all oxygen-loving organisms such as you and I are exposed to. But there's a less understood role of manganese in biology, and that's its role in the immune response. There have been some preliminary studies that have been done in mice showing that manganese can influence susceptibility to infections from a wide array of pathogens, including both bacterial and fungal infections, but no one really understands why manganese does this. In this case, manganese is important for the so-called innate immunity, and that's the body's immediate attack on pathogens, as opposed to so-called acquired immunity, which happens later with antibodies. Now, the worm, C. elegans, has a very simple innate immunity, and this shares many features with the human innate immunity. And our preliminary studies, work that was done by Julie Gleason in the lab, has shown that manganese plays an important role in the worm infection by the pathogenic yeast Candida. So because of this, the worm gives us a perfect tool to dissect exactly what's going on with manganese. We can control the genes in the host, meaning the worm, and we can control the genes in the infectious agent, naming the yeast, and using this we can completely tease apart the mechanism by which manganese impacts innate immunity. And then our hope is to extrapolate what we learn in the worm to bacterial and fungal infections in humans.